Thanks the OBS. Thanks OBS for inviting me. I just get to know that I'm the first one invited to have this cell biology seminar. Really my honor, and it's my pleasure to share my work with everyone here today. It was a very recently published articles that done by our group. So in this study, we are looking at the skin epidermis, particularly the epithelial cells. So I guess without me further strengthening, everybody should know the importance of the skin. And it's good to keep in mind that the skin epidermis is not exclusively to only mammals such as humans, but also to diverse organisms. And skin has served a similar function across all organisms. So in order to maintain the primary function of as the outermost protective barrier, the skin epidermis will have to maintain their skin integrity at all times. Because if there's any serious breach to this structure of skin epidermis, it will possibly causes unwanted consequences, which you do not want to have it. So, but there's already a lot of studies available to address how these skin epithelial cells behave during the skin epidermis development to form a fully functional skin. But still one question remains understudied. And from here, we can see that inevitably almost all organisms will continue to grow in size. So does the skin epidermis. The skin epidermis will have to respond accordingly when an organism grows in size. But we do not know how do individual skin cells collectively behave to support the expanding body surfaces. And this is also the question we wanted to address through this study. So to address these questions, we have chosen the zebrafish larvae as the model organism to study. Well, obviously, it is very small in size. The zebrafish larvae is really small, it's less than 1 centimeter. And because of its small size, it's totally feasible for us to perform a whole animal tracking, which is hardly achieved if you are using a mammal model such as mouse. And it's flat, and most importantly, it is optically transparent. And these advantages make zebrafish larvae as a very good model organism if you wanted to pursue an imaging-based study. And another important criteria for us to choose zebrafish as a model organism, because the zebrafish skin actually do share a lot of similarities with the human skin. So from this, you can see that both have the multi-layered structure. And there are also these basal cells that reside on the basement membrane, serve as the stem cell population that continuously proliferate and contribute to this upper epithelial cells. There are, of course, differences between the human skin and the zebrafish skin, which is that the outermost human skin is covered by the keratinized layer, whereas the zebrafish outermost skin is covered by mucus. So from previous study, we have known the development of the zebrafish skin epidermis is actually very similar to the human skin epidermis. In a very early embryonic stage, the basal epithelial cells serve as the stem cell populations, so they can self-proliferate to populate the stem cell population. At the same time, they are also differentiate to form the superficial epithelial cells. And these superficial epithelial cells are regarded as the terminally differentiated cells, which means they have mostly lost their capability to proliferate and divide. So as an organism continues to grow in size, the skin epidermis will gradually thicken, forming a multi-layered structure in the adult zebrafish. So in order to study the skin cells, we need a very appropriate tool. So in our study, we have integrated the brain-bow technology. Actually, this brain-bow technology was first introduced back in 2007 by this amazing group of scientists. Originally, they used this brain-bow as the name indicated. They used this to study the brain cells. So then my supervisor, Dr. Chen, was very obsessed with this technique. And he has really paid a lot of effort to try to integrate this brain-bow system into the zebrafish model. So if you have followed his study in 2016, he has successfully created this skin ball that specifically labeled the superficial epithelial cells here. And he has studied, using the skin ball, he has studied how the skin's cellular behavior is during the skin homeostasis as well during the skin regeneration process. And when he came back to Taiwan, he brought this skin ball technology with him. And now our lab are mostly focuses on using this multicolor labeling approach to study various types of cells. So actually, the multicolor system is pretty straightforward system. So we have this in each cell we have this multiple copies of this cassette. And if you can see in this cassette, it consists of 3 fluorescence proteins: the red fluorescence protein, the green, and the blue fluorescence protein. And if you still remember your art class, you can simply imagine that by combining different ratio of this red color, green color, blue color, it will create a range of different colors, which you can see from our cells. So this is basically a Cre-Log system. So after the Cre recombinations activity, each cells here will now carry different ratio of this red, green, and blue color. Therefore, they have this very unique color that serves as their barcode. And this fluorescence protein is targeted to the plasma membrane, and therefore we name the fish as the pumpkin, the title here. So pumpkin is not only appealing to watch, but at the same time, it's also a very powerful tool. So you can see from the background here, you can identify each cell. So these pumpkin are able to provide us a very high throughput and high resolution due to its single cell resolution. And because it's a genetic labeling, therefore we can, just like any other lineage tracing study, it can help us to track the changes in each cell across different time points to observe the cellular dynamics within this population of cells. At the same time, coupled with the small size of the zebrafish, we are able to perform a whole organism level, a long-term tracking, which have not yet been achieved in any other vertebrate model. And to be able to have this high throughput analysis, we have collaborated with Sanders. So he has helped us a lot in developing the analysis pipeline to enable us to track the changes of each cell here. So through screening, we have identified pumpkin that only exclusively labeled the superficial epithelial cells, which is the outermost epithelial cells of the skin epidermis in the zebrafish. So from the images here and our quantifications, we can see that more than 80% of the superficial epithelial cells are labeled with a different colored, and it's their unique barcode. So by using this pumpkin, we are able to simultaneously track more than thousands of superficial epithelial cells on the zebrafish larvae. Next, we pursue the tracking. We wanted to know what happened to the superficial epithelial cells when the animal grew. So from 8 days to 10 days, you can see that from the quantification, the animal has already grown a lot in size, with 22% increase in the total surface area. And by doing confocal imaging, you can clearly see that at 10 days post-fertilization, when the zebrafish larvae are bigger in size, there's more cells present as well as the same colored cells. There are more same colored cells indicating a clonal expansion. We then perform a 12-hour time-lapse imaging to see where the cells come from. And from this 12-hour time-lapse imaging, we identified that the cells increase the cell number is actually through the cell division itself. Furthermore, we also proceed a very thorough analysis whereby we track the same animal across 9 days to see what's the ultimate outcome of these divisions. And we found that One superficial epithelial cells, they can divide twice, maximally producing only 4 progeny cells. But just a recap of what we have known previously about the superficial epithelial cells. So in a lot of previous literature studies, this superficial epithelial cells is a widely accepted consensus that they are the post-mitotic cells with little to no proliferation. But what we see from the videos, the time-lapse images, and the long-term tracking is totally a different site, where we can observe they undergo a very massive division. So how can this happen? We then kind of want to reconfirm this. We perform the EDU incorporation assay to detect the DNA replication. So we examine across 4 different time points, collectively more than 10,000 of cells. What we see is that the superficial epithelial cells, there is no EDU signal, indicate that there is no DNA replications activity, as contrast to the stem cell population where we still can detect the DNA replications. So this is quite surprising to us, and then therefore we have this very brave hypothesis that these superficial epithelial cells undergo cell divisions without DNA replication. So we need more evidences to really say that. And it's very natural to, first question that come in mind is that if there's no DNA replications, what happens to the DNA content when it divides? And we hypothesize that the DNA content will reduce. So to really say that, we carry out an experiment to look at the DAPI intensity as well as the H2B, the histone marker fluorescent intensity. So this DAPI intensity and the BFP2 intensity is directly correlated with the DNA content of the cell. So we can see that when the cell that has divided, the DAPI intensity as well as the BFP2 intensity is proportionally reduced, indicating that they have a lower DNA content. And furthermore, we pursue DNA content analysis using the flow cytometry that allow us to simultaneously examine more than 10,000 cells at the same time. And you can see that the red box here indicating sub-2N population, and this sub-2N population are those cells that have a lower DNA content than the usual type diploid. So we can see that the sub-2N population increases from 8 days to 10 days and peak at 14 days, and with almost 50% of the SEC population has a lower DNA content. And this population gradually decreased to almost undetectable in the adult stage. And it's worth to mention here that the 4N population here, it's almost undetectable across all time points, means that the possible of endoreplication is very low. So from here, we are suggesting that the superficial epithelial cells with all the DAPI intensity, H2B fluorescence intensity, flow cytometry, all this evidences suggest that the superficial epithelial cells are able to undergo a very unique cell divisions without the need of the DNA replications. And therefore, we name this alternative cell division as asynthetic fission. But actually, the asynthetic fission, it's not a very perfect system because during this division, we also detected a lot of these error-prone processes, which rarely see in the normal cells. So next, we wonder, what is the purpose of the asynthetic fissions in the SEC? And during this tracking period, we found that the zebrafish grow the most rapid from 8 days to 14 days. And this coincided with the timing of the asynthetic fissions. Another interesting observation is that the cell that has undergone asynthetic fission, that appear overall smaller with a reduced apical surface area, reduced cell thickness, as well as reduced cell volume. In this observation, we have collaborated with our with Sanders and as well as Cherry to help us build this simple geometric model to explain the findings. So in this simple geometric model, we assume that during the asynthetic fission, there's no need to synthesize new materials where only the existing cellular components, including the plasma membranes and cytoplasms, are utilized to construct new cells after each division. And through simulation, can see that independent of the cell shape, no matter what shapes they are, during the asynthetic fission consistently in their first division, there's a 26% increase in the total surface coverage and 59% increase when they have divided twice. And this simulation data is further supported by our experimental data with a closed fit. With this geometric model, we propose that the asynthetic fission is a very frugal yet highly efficient strategy to provide more surface coverage when the zebrafish has the most rapid expansion. So if this holds true, we can expect that the frequency of the asynthetic fissions is actually regulated by how high is the surface demand. So we have this very fun experiment whereby we rear the zebrafish at different density. By doing so, we can manipulate their growth rate, therefore creating a scenario for different surface demand. So for those fishes rearing in a high density, they will grow slower from this quantification. When they grow slower, they appear smaller, therefore lower surface demand. And vice versa, when they are in a low density, they grow faster, result in a bigger size, therefore a higher surface demand. And indeed, in this condition, we can see that the frequency of the asynthetic fissions is higher when the surface demand is high as compared to those fish that has a lower surface demand. Next, we wanted to know what triggers the asynthetic fission. And during this period, the zebrafish larvae continue to grow inside and it's the most rapid phase of growth. So we reason that during this growing period, there's the tensions built up. And through literature, we have targeted a very particularly interesting stretch-activated ion channel, which is the piezo1. And this piezo1 can act as a homeostasis sensor. It has been shown to involve in live cell extrusion as well as cell division in the epithelial cells. So first, we do a drug treatment. By treating them with the tension inhibitor, gadolinium, we can see that the cells rarely undergo asynthetic fissions. But when we activate the tensions through treatment of YODA1 that's specific to this piezo1 channel, the asynthetic fissions increase, result in a larger clone size as well as a more divided pole here. Further, we also validated this data with a CRISPR-Cas9 approach, whereby we mosaically knock down the piezo1 in the palm skin and examine the clone size as well as the divided pole. And we can see that by knocking out the piezo1, the clone size decreased as well as the divided pole decreased. And this part was helped by Alan. Actually, I saw his name in the, okay, hi, Alan. So just to sum up our study, we have identified a unique cell divisions, alternative cell divisions that happens on the superficial epithelial cells. And this cell division, we name it as asynthetic fission. And this asynthetic fission, the special of this asynthetic fission is that it can proceed without the need of DNA replication and therefore result in a lower genomic content. At the same time, it serves a purpose. The purpose of the asynthetic fission is to increase more total surface coverage when the demand is high, especially during this rapid growth phase. So actually, these studies has already published in Nature. So if you are free and you wanted to know more, please feel free to read it, because I actually didn't discuss a lot of details here today, just a sharing session. So I do not focus on too much on it. I just want everybody to know the take-home message of this story. So last but not least, I would have to acknowledge all these amazing people, the team behind this work. especially my supervisor, Dr. Chen Hui, has guided me well throughout my PhD study. Xiaoyu has helped in some of the experiment. Alan helping me to for the CRISPR-Cas9 generation. And Cherry and Sanders, our collaborators who help us in the mathematical modeling as well as the analysis pipeline development and other core facility staff. Lutu and Peggy helping in some instrument settings as well as some analysis. So if you have further questions or anything, you can feel free to contact me anytime through my Gmail. And with that, thank you.
Welcome. Thank you, Keat, for your presentation. That's very interesting, your work, and how you describe a new type of cell division. I think that this information is breaking news for all the biology, all the biology, and this will revolutionize what is in the books, the textbooks. I have a question, and I will open this time of questions for you. You have fishes in different population density. You have low density and high density populations. And you think you saw differences in the length. And do you think that there is also a difference in the number of in the presence of this type of cell division? And also, what do you think that is a mechanism that that induces this kind of division in response to the density of the population? Do you think that there is a codification in the by the nervous system of the fish, or do you think that this channel that you discovered that is present in these cells, that is sensing on these cells the composition of the skin? Do you think that there maybe there's a mechanism of direct contact between fishes that are inducing locally this kind of division?
Sorry,
sorry.
I don't really get the questions, but if I'm not wrong, are you asking what's the mechanism that causes these asynthetic fissions to occur when they are varying in different density?
Yeah. Do you think, do you have a hypothesis of what kind of mechanism are inducing this kind of division depending on the density of the population?
Yeah. So in our study, we actually identify these stretch activated ion channels possibly involved in this asynthetic fission. But I have to admit that these are most probably not the only channel that involved in asynthetic fissions. And of course, there are still a lot of like direct regulators waited to be discovered. But from the different density experiments, because in this different density, we can see that the cell size, but not the cell size, the fish size is also different. So in the low density, the fish size appeared to be larger. So when they are larger, we can assume that the larger surface area probably provide more surface tension, and this surface tension possibly could act as a key to trigger the asynthetic fission. Does that answer your question?
Yeah. Okay, but the fish detect this different density by independent mechanism. It's a global mechanism that is.
Yes.
Okay,
yes.
that induces differences in the growth, and this difference in the growth induces differential skin stretching forces.
Yes.
Okay. Yeah. Oh. Okay. Well, that was my question. Thank you for answering. And so on. Anyone have question. I see
this is Guan. I think the host unmute me, so I'll go first.
Okay.
I think I see many people with questions. So that was fascinating, Keat. Thank you for sharing. I'm wondering, do you see this as a conserved mechanism across different families or organisms, like for example in humans?
That's a very great question, actually, because we are the first one to discover such a novel cell division. Actually, definitely still a lot more work to do. We are also collaborating, hopefully, by opening this new field of study. Hopefully, other people that using different research model that also can join in to see whether this is a conserved mechanism across all these animals. Yeah, I have to say this is yet to be discovered. I do not know yet.
I see. Okay, then I suppose the second question is related, so it probably doesn't have answer yet. I wonder if things like cancer use similar mechanisms to expand their reach and expansion too.
It could probably, yeah. I also don't have a definite answer for this. It definitely need more investigation on it.
Thank you so much.
Thanks for the question. Well, those are very, very good direction that we can go to in the future.
Sure, thank you for your answer. Well, we have hands raised. I will. Yeah.
Okay. My name is Jinghua Shi, and interesting talk. So I have two questions. One is have you measured how long would the cell generate by the asymmetry division? Because I'm thinking about if those cells are just like a temporary plant, so that just responding to the environmental change, and later when those cells die, they will generate all the nuclei, cell with a real nuclei. And the second question is, you use the H2AZ and cherry to detect the division. Since the H2AZ, not H2AZ, H2A and cherry, since H2A has a lot of different variants, I'm wondering how precise that thing can target, because H2A variant is very important for zebrafish in different pathway. So just wondering how precise they can detect. Yeah, that's my question.
Oh, okay. So go back to your first question. How long does this cell stay on the zebrafish body? Actually, the zebrafish skin also has a skin cycle, just like in mammals. So basically, these superficial epithelial cells, they will eventually shed off in about a month. So when we track up to 21 days, this superficial epithelial cell that has undergone this asynthetic fission actually all shed off and repopulate possibly by the basal stem cells through differentiation. Therefore, that's why we speculate that even though this asynthetic fission, the DNA content may not be normal for this cell, but they eventually shed off anywhere, so they do not cause harm to this organism. And the second question, are we using the H2A to perform to conclude that these cells undergo asynthetic fission? Actually, it's a H2B version. Well, of course, we have different evidences to say that the DNA content reduced. For example, here we stain with DAPI staining, and we also perform flow cytometry analysis. So I guess with this accumulative evidences, we are quite confident to say that the DNA content reduced during this asynthetic fission, and it's possibly not because of the using of different variant of this histone markers. Am I clear?
Well, thank you for your answer. I will continue with the next question is from Chung Yao. Go on with your question, Chung.
Yeah, cool. Thank you for your talk. It's very interesting. Just wonder for the mechanism part, like how exactly is this process undergo? Because like does it still form a metaphase that chromosomes have to align? Because without replications, the cell cycle checkpoint will not be able to proceed if it's following a normal checkpoint mechanism, right? Because you do need chromosomes to align and then such that the cell division can occur. So I just wonder what kind of proposal you guys have for this kind of detailed molecular mechanisms by which the chromosomes, even though they are not replicating aligned, they can still separate.
Yeah, that's actually a very interesting question. That's also like currently we are still progressing this way. We also wanted to know how this chromosome without pairing, how do they actually segregate? Oh, this is, there's still a long way to go to understand how it happened. But in these studies, we have actually used different drug inhibitors that inhibit a different cell cycle checkpoint during the cell cycle. And we see that by using the drug, there's no effect for this asynthetic fission, which means the asynthetic fission can still progress normally, even though we treated with this inhibitor. It's just that when we inhibit we use the nocodazole, which inhibit the tubulin formations. It affects the chromosome segregation, and we can see binucleated cells. Other than that, if the other drugs, they do not inhibit the asynthetic fission, which means possibly the asynthetic fission, the cell cycle machinery, probably is totally different thing than the normal cell cycle that we know. And I would say it's still a long way to go to fully understand the full picture of this asynthetic fission. Yeah, hopefully we can find out one day in the future with everybody's help.
Okay, thank you for your answer. Now we have Hsing.
Yeah,
Do you have
it's me
another
again.
question?
Yeah, it's me again.
Okay, go.
So you did experiment with the different density, different population density. So I'm
Yes.
wondering if you change the density over time, would that phenotype, the asymmetric division will change over time based on like what I mean is like, would that asymmetric division is the once they set up, there will be always that, or just like they will sense the environmental change all the time.
Oh, well, we did not do any experiment, like, for example, like from low density, we change to high density, we did not do the experiment. But from my understanding, let's say if the fish has already grown big in a low density condition, and it has already big, and the asynthetic fission has already occurred. So even when we move back to high density, yeah, I would say it probably wouldn't affect much on the asynthetic fission itself.
Yeah, because the reason I'm thinking about it is because there's every kind of insect, they have the windy form and windy form, and they will change it the wind side with the wind or not based on the population density. So if you move to the low density, they will start to lose their wind, and if you move back to the higher density, they will start to generate wind again. So I'm wondering, would that change over time as well?
Oh, that's still a very cool experiment to try on. Yeah, but right now, because we have not done it yet, so I probably could not provide a very perfect answer for this.
Okay, thank you.
Thank you.
Well, is there another question for the public? From the audience, another question? Well, we have a question from Eugenio.
Hi, thank you, Keat, for this great work, and I'm really happy to have you with us today. So I want to ask you just a really few questions. The first one will be, what is the basal transcription rate of this cells that does not have complete chromosomes?
That's actually a great question too. Well, we are pursuing this to check whether the transcription, the basal transcription, if there's any changes in this cells. It's still currently under work, so I probably could not provide a very a perfect answer for this yet.
Okay. And my second question somewhat related, but if you have any sort of idea what's going on with the mitochondrial dynamics, because we know that during cell division we have the same rate of mitochondria. Is this happening the same? Do you have any sort of idea? And my third question will be the cell death mechanism of these cells, because we know, for instance, that during failed mitosis you have mitotic catastrophic cell death. So what's going on with this cell death? If you are looking for that, what will be the next direction of this research?
Well, regarding the mitochondrial dynamics, well, not only mitochondrial dynamics, you can just, in one cell, there are a lot of organelles inside. I guess it's pretty interesting to see, besides seeing how this DNA is segregated, it's very interesting to see how this organelle divide into two cells, given that there's probably no enough genetic materials. So we are still currently pursuing this. So we actually have already fostered some collaborations to help us look at how this either the RNA or protein, how they are actually distributed in these cells after asynthetic fission. And the second question, may you repeat? I kind of forget.
Yeah, like the cell death mechanism.
Cell death, yeah. So in our study, we actually have evidence showing that they do not undergo apoptosis because we have just did not detect the apoptosis. So we reason that the cell just shed off from the surface. It doesn't undergo like classical cell death such as apoptosis or necrosis. Yeah, but it's still a possible direction that we can look at them. Maybe they are undergoing some very special cell death program that enable them to shed off from the skin.
Well, thank you very much. I guess we don't have more questions. So thank you for Keat for your presentation, and I'm really happy to have you as a first presenter of cell biology seminar, which I hope we have a lot of in the future. And I put in the chat box our links of our social networks, and please feel free to fill the form. I will put it here once again, so you could we could have you an idea of what the topics you would like to hear in our future symposiums, and follow us for our next sessions. Thank you very much. Thank you, Manuel. Thank you, Keat, and congratulations, everyone. Thank you. Thanks for joining. Thank you. Bye-bye.
Thank you. Bye.